All right. Hey, good morning, everybody. Craig Davidson here, Taurus Agricultural Marketing. Welcome to another version of our Taurus Agronomic Bites. This is a continuation in our series of what we have called the 100 Bushel Club. You know, our last podcast was Ken King and more focus on canola. Obviously, a great conversation. Today, we're looking to extend that with a little more emphasis on pulses and what we're calling the next human protein crop. We're glad to have with us this morning Meghan Rose, New Era Ag-Research, and Ryan Immerkar with RSI Consulting and New Era Ag, as well as Mike Dolinski, who's been a part of most all our podcasts, and glad to have him on here again this morning. I'll maybe let Meghan and Ryan introduce themselves, and then we'll actually get into our conversation this morning.
Thanks, Craig. I have to admit, this is my first podcast, so I got Kelly to get me a fancy headset here. I feel like I'm working at Hortons, maybe. But,
you look good.
Yeah, thanks. I'm owner of RSI Agri-Coaching. I started with AgriTrend over 10 years ago, and on my own now, consulting. 7 years ago, I partnered with Dennis Badowski. We started New Era Ag Technologies. It's independent retail, and with that, 2 years after starting that, we found Meghan, and we started New Era Ag-Research, and it's been a lot of cool and exciting things happened since then, and Meghan's business and the retail have grown exponentially over that time, and we started the retail research business for the reason of just trying to explore and understand products on the market and what's possible in the Swan River Valley, like for our growing area. That was the main reason for starting it, just to understand how these products worked in the Swan Valley, because we all know things work a little different everywhere across the prairies and the world. So we've learned a tremendous amount, and it's snowballing for sure. Meghan can maybe talk a little bit more on when she introduces herself on what we're doing and things that we're doing and what we're doing. So yeah, go ahead, Meghan.
Happy to be on the podcast, and this is a new thing for me as well. So New Era Ag-Research, as Ryan said, started in 2016. We do both small plot research trials as well as field scale, which I'll show you guys or talk a little bit about some of the research that we've done in peas and fabas over the years. We work in not just pulses, but 9 different crops from hybrid fall rye. We do a lot of work in quinoa, canola. We work with 9 different breeding programs in screening their yield trial, their advanced yield trial material. And we've just been kind of growing since every year. There's more projects to do. So I think what I've learned in the last few years is how it's really important to do on-farm trials, and collecting information for our valley is really unique. And not only is there differences just for our valley, but for field to field, farm to farm, and doing your own on-farm trials is really valuable. So, yeah.
Yeah. Fantastic. Yeah. You know what? I mean, it's never too late to learn something new. So for Ryan and Meghan, glad this is your first kick at the podcast world, and we're glad to have you on. So, I would actually, we appreciate the feedback too. So if you're on watching or listening, you do have an opportunity to ask questions throughout, and we'll try to get to those as we go through our conversation. If we don't get to them through the conversation, we'll get to them at the end, or if we can't, if we run out of time, then we'll actually follow up with an email with answers to the questions that you had asked throughout. So obviously today we have a very technical panel, obviously. So, I'm a podcast guy. Mike likes his webinars. So I call this a webcast. It's kind of somewhere in the middle. We got quite a few slides that we're going to show too, because it's, we're actually dialing in. We're going to talk about peas and Fabas and in the 100 bushel mindset. And it's definitely there. It's real. And, this is today as the conversation is about, what do we need to do? What are some of the things we absolutely have to look at if we want to focus on these types of target yields? And what are some of the challenges or limitations that's probably going to hold us back from actually getting there? So buckle in. Good conversation that we plan to have, and yes, there will be some reference slides here today to give you a little bit more information as opposed to just general conversation. So I'll start with you, Mike, and when you lay the framework, you get, you've been in this business for, I'm not going to date you, but you've been in this business for, call it, decades. How you probably get piles of questions when it comes to crop production. When you look at things like pulses, specifically peas in this case, what are some of the aspects that you're going to say we need to bring into consideration to focus on, a high target yield crop here?
Well, the one thing about peas that is probably to their downfall is they have a relatively small rooting system compared to many of our crops. They don't go down very deep, so they're mostly focused in that top foot, like many plants, but they don't have a lot of depth in many ways, and they're very sensitive to salinity, to tell the honest truth, and they're also very sensitive to toxins in the seed row, because they have a very thin seed coat, and both the seed coat can be damaged. So I'm a big believer, like in many crops, it's a total balanced package of nutrition and management. So for some of these that to me are key are always seed quality, and this seeding rate, and I think maybe Meghan can talk about this, and of the data such that somewhere around 7 to 9 plants per square foot is key, and good quality seed is really important. All of these factors here are not going to be new to folks on this. Maybe go back, Craig, for a sec on the yield. I'll just cover those. pH, just a few quick comments for guys. Maybe it's worthwhile spending a few minutes on this, because a lot of guys, maybe in Manitoba, with the development of the pea protein extraction systems that are coming on, and the industries that are developing, are going to be maybe first-time pea growers. So pH is key. They don't really like high pHs, to tell you the honest truth, in many cases. And I mentioned salinity. They don't like salinity. They don't like wet feet at all. Peas do not like being submerged in water for any length of time. And it's also been one of the major causes of phenomyces, which is a real nightmare for that whole pea lentil regime that is going on. So for me, the key ones are basic agriculture: good quality seed, proper seeding, proper nutrition. And nutrition is always the problem for most of our problems, or most of our crops. Guys are cutting back. We're producing more yield per acre at a faster rate. Plants are having to pull more nutrient faster, and we're depleting our soil slowly over time. And that's a real problem as far as I'm concerned. So I think when we get into the details, it'll be more important. There is one key thing for new beginners on peas. Peas take a lot of water to germinate. They're a big seed. It takes a lot of water to germinate that seed. And one of the key problems you can face is if you don't seed deep enough, and you get partially through germination and the plant runs out of water, that's a serious problem. So with peas, you can seed them quite deep, because the seed stays below ground. It doesn't move the cotyledons above ground. So most guys seed early because they're quite frost tolerant, and if you freeze off the top, they'll branch out. But the seed remains below ground, so it's quite tolerant to severe frost. But if it partially germinates and doesn't get fully imbibed with water, it's a real serious problem. So the general rule is seed to water. Seeding early is good, but you don't want to really seed it into dry land as far as I'm concerned.
I would say, Mike,
we'll talk about later, but go ahead, Ryan.
I would say, Mike, too, if I was to prioritize on that list there, I know some people might argue this, but next to water, my next thing, low-hanging fruit that really affects yield is weeds, I find. If you don't have a proper, they're such a poor competitor at the start of their life. If you don't have a clean field, or you don't keep fields clean, you don't do a pre-seed burn-off and a post-seed burn-off, these are easy things that you can control that make a big difference to start your year off. If you have a bad weedy field, dirty field, you could do all the fertility work you want, and it's not going to produce the way you want. You're going to be disappointed. You need to start off right, like you said. Obviously, a stand is great, but the weeds will affect the plant stand as well, right? Like and how that grows. So I've noticed over my years scouting these fields, if you can't, don't pick a field with tough to kill weeds. Like, you're limited a little bit on what you can use for chemical in pulses too. So keep that in mind. If you have real tough, heavy buckwheat and things like that, these are things that are dandelions and weeds, or perennial weeds. It can have a huge effect on yield right out of the gate. So just wanted to make that point.
So you're, I guess you're probably alluding to rotation in that mindset too, then, Ryan, as part of a strategy.
Yeah.
To manage or control weeds.
For sure, and there's all sorts of products in the market, but everyone's weed spectrum is a bit different. But I think if you don't be afraid to spend money on chemical with peas, for sure. Like whether it be Edge or a post seed of heavy rate of heat, or there's lots of different options. But I would say it's money well spent when it comes to most pulses. So.
Yeah.
This is a good slide to summarize all this for people who are just on the podcast. This will be posted. There are a few slides here that sort of outline all the key ones, and that one is. I'm glad you brought that up, because the trick with any plant that nodulates, a soybean or any of the legumes, besides being tough to get going early, they run into a nutrient deficiency and a growth sort of problem, because when they're starting to nodulate, they're using a lot of that energy. So if you don't have the weeds controlled, the weeds will overrun them, because there's a period of time where they're slow growing as they're building those nodules and growing all that vegetative period.
Yeah,
it's really critical.
Yeah.
Good point.
Yeah, we'll maybe come back to that. We got a slide that we'll talk to that specifically, and Meghan's done some research on weed control in peas, so I'll maybe let her mention that here quickly.
I didn't do the research myself, but I did do a little bit of Google research, and the Saskatchewan pulse growers had some good information about the critical weed-free period for peas. It's not established for faba beans, which I was surprised, but I think we can kind of extrapolate it would be similar, being that they're also a pretty low competitive crop. But 1 week after emergence was when the critical weed-free period started, which is that's pretty quick. Like, you're not probably going to be going in there with your in-crop herbicide application 1 week after emergence. So to have something that's clean from that point, to have a clean field from that point on is important. So
there's a question here while we're on the topic. As a pre-seed emerge, what chemistry would you look at? Or what have you used in your experience, Ryan, for peas specifically?
Well, Edge is expensive, but it does work well, and it doesn't require a rainfall to activate it most times. It does help, but I find a heavy rate of heat works great. You just need a little bit of rain. I find it hangs on longer than you'd expect too. Like the photodegradation isn't as bad as they might claim, but it certainly helps with volunteer canola. We have a hard time with that here in the valley. Like, it's a terrible weed in pulses right at the start, because it germinates so quickly and it can take over so fast, and you're limited to the amount of times and the timing when you can spray Viper and different products like that too. So It's not a Liberty or a Roundup where you can just keep nailing it over and over again and that kind of thing. So, I would say my advice is just, I don't really know the weeds that you're dealing with, but I guess the takeaway is don't be afraid to spend money on weed control, post and pre-emergent products in peas. It's ROI is huge when it comes to growing pulses. It's the best money you'll spend. Next to fertilizer for sure, or I would even claim before fertilizer. If you or just don't pick a proper field too. Again, don't pick a field full of wild oats and Group 1 resistant wild oats for sure, right? Because you're hamstringing yourself again right out of the gate. So
yeah, I would say, this is a good conversation around weed control and chemistry. I would put a caveat in here in my mindset, because essentially we are talking about human food here at this point, right? I mean, we are, you think of the history of pulses and how it's grown. I mean, lentils have always been that case, but today coming to North America, and we're talking now, we got pea processing to human food, either in sports drinks or, stuff that looks like meat or whatever. But I think we're going to see a lot more critical eye on how these crops are actually being grown and produced. And then in things like chemistries, because we've seen it in other crops like oats where, we can't spray glyphosate as a harvest management tool. I'm going to question moving forward, what are our options going to be when it comes to chemistries, if in fact these, say, pea crops are growing for protein, but ultimately for human consumption. And that's something we got to be cognizant of as agronomists and people in the industry moving forward. Absolutely. Which is also another great challenge that we're probably going to be presented with when, as Ryan mentioned, how important it is to manage and control weeds early. Yeah.
There's a lot of
Meghan,
some protein as well in our peas as best we can. And I think you did some work on that we'll probably get to later.
Yeah. One quick question, Meghan, when it comes to your research plots, and in Mike's slide, and if you can't see the podcast, obviously Mike's referencing a wide range of seeding depth, anywhere from 1 to 4, but basically focus on chasing the moisture, which is good for peas. And your research plots historically, how deep have you been seeding your peas?
We're going about 1.5 to 2 inches, trying to stay in that. Obviously, you want to hit the moisture, and then once you roll it to, I don't know if it's our opener or how it is, but we always seem to get a lot deeper once it's rolled. So I'm hesitant to the actual seeding depth when we're checking it to go too deep, just because we do have that extra crust to get through. And I always question that too when we're talking about seeding depths with soybeans or peas, because what is that at the time of seeding, or is that after you roll it? Because that can make a huge difference. So I've never had an issue chasing moisture with peas. Usually we're in early enough that we don't have to go too deep. I find it's a problem when we're chasing the moisture for soybeans. Then that's a different issue, but yeah.
Most of the time it isn't a problem because guys seed peas so early, so there is whatever moisture is there is generally there in the upper surface, and you're right, you seed as shallow as you can because you want to get it up and growing. For guys that are looking at this issue, I mean, potassium becomes very, very important to any growth in the plant in terms of hydraulic pressures that it puts on to grow that seed. So if you're going to seed deep, you want to make sure your potassium levels, in my opinion, are pretty adequate in that top zone. And in most soils where you get minimum potassium levels in the top 3 inches are pretty adequate. So, the peas are in a good position to grow.
I have a reference here, and if you can't see it, but this is nutrient concentration in seed, and this is something that we have a lot of emphasis and focus on right now at Taurus, and Mike's doing a lot of work on it. And we believe, the day you put your seed in the ground is you can never get that day back. So you have to start with a very high quality seed. But we also believe there's a direct correlation to nutrient concentration of the seed and how strong or how vigorous that seed actually is. And some research we're doing right now with 2020 and Bayer and looking at some other seed lots is going to help us define that. But one thing that jumps out at us when we look at peas, obviously a very high seeding rate. Most growers are going at 3 bushels an acre. Is that fair, Ryan? Is that what most of your recommendations are at?
Depending on 1,000 kernel weights, but we try and target that. Yeah, like it'll be between 3 and 4. Some of these seeds are getting pretty big. Like there's some varieties like Abarth, they have like jumbo seed, and the seeding rate does go up to try and get that desired plant count of 8 plants per foot. So there's a big difference. I think a lot of that yield with the Abarth does come from the size of that seed. It is significantly larger on some varieties. So it's just funny that I think they're finding that the protein on some of these varieties are, which does kind of make sense, are a bit lower too. They might not be the most attractive for these protein plants. So I think that goes hand in hand with anything that you're looking for protein, like wheat yield and protein as well, right? So it makes sense.
Yeah. The higher the yield, the harder it is to bring the protein along with it, right? So it's I would argue, you talk 4 bushels, call it a 60-pound bushel crop. 4 bushels, you're talking 240 pounds of material, and you could be putting on more pea seed than you are nutrition, which is crazy, right? It's
some pretty big eye rolls when it comes to trying to recommend that from customers. It's kind of like then you want to get into faba beans as well. If we can get some huge faba bean seed sometimes, and it's almost like you said, logistics trump agronomy. Sometimes you just can't get that seed on. It plugs the drill, and it's just kind of can be a nightmare seeding, to be honest. So
a quick point here. Obviously nutrient concentration in the seed is important at that seeding rate. I mean, I think that we did the math at one time. You could be applying as much as 8 to 10 pounds of phosphorus an acre just in pea seed alone, with the phosphorus concentration in the seed. So you talk about nutrient importance in the seed, peas especially, because your seeding rate, if you're low in one of these nutrients, phosphorus, it loads a huge amount of potassium into the seed coat. So I would say we're talking 100 bushels here. To get there, I'd say let's start with actually doing a nutrient analysis of your seed. I think that's important, because if it's actually short, we have agronomic tools to shore that up. I mean, we can add nutrient dressings. It's not like you're left in the dark here. You say, well, if it's a little bit low, let's shore that up before we actually even put it in the ground.
Right. And I think you have to be aware of, seed place potassium with most pulses is not an easy thing to deal with. You shouldn't do it, actually. The potassium chloride and they're not a good mix. So you're looking at other ways, side banding or broadcasting.
The salter mix of potassium chloride is one of the big reasons why it doesn't fit well in a seed row with a something like peas.
Right, and I think I worry a little bit about the inoculant too as well, how the survivability without that salt in the seed row too. So
so you make a great point, and kind of the next phase of our conversation here is really to talk about nutrition and fertility and the demands that this crop has. And this is you brought it up to a T here. I mean, very high, and I'll show you this, very high nutrient using crop, one of the highest we'll actually grow in our broad spectrum crops in Western Canada. And yet inevitably we run into this conversation on seed safety. And I truly believe peas, no different than soybeans, they are a crop that absolutely loves fertility. They need a lot of fertility, but they really don't like fertilizer. They don't like fertilizer because fertilizer is basically nutrients with salt. I always mention this. So I think it's part of our responsibility. If we're targeting 100, we've got to make sure the fertility is either in the soil or we're adding fertility as nutrients, but not adding fertilizer that could actually harm or injure this crop early in its days. And phosphorus is an issue. You mentioned potassium. It's such a huge potassium user, but it doesn't like potassium chloride. So what do we do? Right? We're heavily focused on that at Taurus to try to find options like a crystal green or a sulfur plus, or we're going to talk about polysulfate in the coming months, but options that actually have very low salt index, so we can actually address fertility when we actually plant and grow these crops. And it doesn't stop with, the demand of the macros. I mean, Mike will reference the importance of some of these key micros and actually the overall functionality of a pulse plant, and all the way down to nutrients like moly. And you say, well, who cares about moly? But, it actually has a direct correlation to nitrogen. I'll let Mike mention that quickly as we move through these talking points.
Yeah. I mean, all of these are important, and a lot of people don't think of it, but how zinc, boron, and moly are important. Moly is involved in breaking down that nitrogen atom. So without the plant, without that, it can't actually fix nitrogen well. But you guys in say Swan River, high pH, where you have probably adequate moly. Moly is one of the micros that actually becomes less available as the pH goes down. So it's a concern in more acid soils. But one of the key ones is boron. Zinc is needed by every plant. A farmer should never let his plants run short of zinc. Never. He shouldn't let his plants run short of any of these nutrients. But zinc is in all the key enzyme groups in the plant and key for stress management, just like us. If you guys are all watching COVID stuff, you know that vitamin D and zinc are kind of the two materials you want to have lots of in your system, not to be toxic, to prevent it. But I'll just give you an example quickly on boron. For example, when a plant is building the nodules, I'm a believer that boron has to be there throughout the life of the plant, and that it feeds through soil. And when you're putting on a foliar, you're sort of touching it up. You're not solving a nutrient problem. But when the plant builds the nodule, and for folks that aren't familiar with that, the plant builds the nodule, not the bacteria. So it has to restructure and build all those cells in the nodule, and that takes a lot of calcium and boron and phosphorus and you name it. So that is why we need a lot of boron right through the plant growth for all the tissue growth and then reproduction. All these micros in legumes are perhaps more important than in some of the other plants, but these are all important. And take a look at these slides when you have a chance here, because it outlines where you have some possibilities to improve your productivity. And we didn't look at it in the last one, but when you look at the seed nutrient content in peas, the levels of boron in the actual seed are far higher than they are in cereals. Canola would also be very high. As a general rule of thumb, dicotyledonous plants use more boron than monocots, just the way the plant grows. So all these micros are important. Now, I sort of focus on micros because we've been farming here for 100 years and we haven't been putting anything back. And you only need to really worry about them if you're short, like any macro. So don't be upset that I'm pushing or think I'm pushing micros. I'm not. You just need to consider them.
Yeah, especially if we look at. I was just going to say, if we look at the pulse growing region in Western Canada, a lot of them are growing in what we'd consider the brown soil zone, and as Mike would attest to, that's an area that is fundamentally deficient in zinc. There's just no way around it. It is low in zinc, and yet we see zinc deficiency in a broad spectrum when we start to get very specific on our soil testing. As Ryan has done over the last 20 years of his career. I mean, I don't have to tell you, but where deficiencies lie across the valley.
Yeah, for sure. And one thing that does, some of these immobile nutrients, they don't change a lot, from year to year. Like copper, for instance, you can soil test from year to year. The number doesn't change unless you do some kind of a major application, bluestone or something. I've seen it actually change in the soil. But once you know. But I find boron, like nitrogen, can change from year to year from a soil test standpoint. I think that it comes out of the organic matter. Correct me if I'm wrong. Most of the boron, like naturally.
That's what's stored, yeah.
Dry weather, it is leachable as well. I find it's even that much more important when it's in dry, droughty conditions for pollination, right? Just because just how pollination works. So it's one thing that can change from year to year, boron levels. And I was just going to just quickly note on that. You guys are probably wondering what attention levels are. Sometimes I find parts per million, 0.2 parts per million, and it's a no-brainer. Those ones are easy to make a decision on. And then we talked earlier about how to apply boron. It's nicer to get it in the soil. It's just one less thing to worry about. When you look at foliar applying boron, it's tough. Again, logistics, guys like to throw it in with their fungicides in canola or peas or whatever, and maybe the timing might not be great for boron because that becomes a bit of a struggle. So it's always a little bit nicer to get it in the soil, but there's ways to put it on foliar as well, but it might just be more of an inconvenience, I guess, than anything. So to try and do that.
Yeah. Not to lose sight of our attendees, we got another question that I want to miss, or kind of just went over it. But Mike, where do we send our seeds for nutrient analysis, and what specific tests are we getting done, just so people know if they want to send their samples away?
Well, I use A&L because Greg Patterson is also very interested in this whole nutrient density issue, and I've used that, and I just do it's the P1 test listed on their program, and it's their tissue test analysis basically. We don't have a seed nutrient density test specific for that, but when they break down the kernel or the pea. It extracts pretty well everything, so that's what we're using, just the regular tissue test analysis. My thesis is that, and I'm seeing this now, is we're just in the beginning of this, is that the seed will tell you. We hope that by looking at the nutrient content of the seed, we can predict vigor and germination. That's what the study's focusing on. But I'm convinced over time that if we do a soil test, a tissue test, a seed analysis, that completes the cycle of the plant. And I think it's going to be the seed test that is going to tell you what the mother plant and that crop could not get in that year of production. And if we do this through a couple of cycles, I think we'll really have a better understanding of not just how to fertilize it, but how the plant perceives that fertility and can utilize that fertility that's put on. And it just coincidental. I mean, I did a couple of webinars for Crop Intelligence, and they're going to start doing some studies tying in all of this into their moisture sensing apparatus in the field, so we can tie water into the whole cycle. So I'm pretty optimistic we're going to learn a lot about this. Just starting.
Okay. Yeah, that's a great study that we're conducting, and it's going to provide great information, especially when we're talking, pursuing higher yields. What are our limiting factors? And thanks for that question, Darcy. We appreciate that, because that is going to be valuable moving forward. I'm showing here total demand for 100 bushels. I've highlighted peas. We showed this slide on the last 100 bushel podcast. Again, if we rank the demand, overall peas, if I added up all the macronutrients, NPK, calcium, magnesium, what does that look like? I say to grow 100 bushels, that pea crop on that acre needs to find 747 pounds of nutrient. Well, we compare that to barley. Barley's 365. But the caveat here again is 300 of the 700 is actually nitrogen that theoretically, if we're leveraging biology, hopefully we can use nature to actually help us find a big percentage of that nutrient. But that still leaves 400 and some pounds of other nutrients, and a lot of that is actually going to be potassium. So it is, you put it in context, you want to grow high yielding pulses, peas specifically, you better not start on a field that has low levels of nutrition, because if it does, chances are, you're just not going to get there. And we'll reference this later in our conversation today with Meghan, as we look specifically at some of her research sites over the last 3 years, and the level of nutrition that those sites have, and what it meant for actual production. So, if you're starting with high targets, you better figure out what you got to work with, because peas as a crop, they have their foot on the gas, and it's not just at the start, it's all season long.
Brian and I were kind of talking about that yesterday, about just, it's not something that you can, decide to do 1 year. You really have to be preparing for a few years and building up that soil. You're never going to I mean, I guess you could. It probably wouldn't be economical though to apply that much nutrition in a single year. So it definitely is a multi-year approach to that.
I find too there's nutrients that are hard to remedy in the soil, right? Like I find N and nitrogen and sulfur seem to be simple to remedy. You have X, you need Y, you just add more and you get what you get, right? Where phosphate and potash, they're tougher nuts to crack a little bit on getting to where you need available phos and potash, so or potassium. So, it's not as easy to remedy.
Yeah. Now the good thing with peas, if in fact everybody is following a rotation due to disease management, you might have 6 or 7 years to build into it, right? If you follow the guidelines, you say, well, I got 6 years to build up my fertility before I get back to peas. But here we are. I'm going to show you another indication of our challenge. We talk about peas and we look at its sensitivity to fertilizer, and I'm talking specifically salt load or salt index. Of all our pulse crops that we would grow here in Western Canada, are the biggest group of them. Theoretically, peas could be considered the most sensitive, and this is work done by Dr. Jeff Shanu at the University of Saskatchewan, where he's telling us no more than 15 pounds of phosphate in the seed row for peas versus Fabas, where you could be as high as 40. So, if you're thinking, what is the right pulse crop? I'm low in phosphorus. Maybe seed safety is part of your factor in deciding which pulse crop to pick. And as Meghan knows, researching both crops, that's always built into her plan when she puts the fertility together. And that's why she's probably broadcasting. Would that be true?
Yeah, you just can't get it. Yeah, we have a mid-row band and side band option, so that works for us pretty good. But yeah.
Well, if you look at putting peas after canola, you've got a couple issues, because canola pull a pound of pea per bushel. If you're going to follow canola with peas and you're pulling a 50-60 bushel of canola crop off, your pea level, you better be starting a couple of years ahead of that pea crop, man, if you're going to try any kind of a stunt like that, because you're going to be short of pea.
Yeah. And this isn't about products, but we are adamant. The future of fertility is bringing nutrients without salt, and that's why we think a product like Crystal Green is so imperative in agriculture. Why it's such a good fit in crops like pulses, because we're letting the crop actually do the feeding itself, and we're taking the salt away, so there's never any injury, to that seedling or the early part of that plant's life. And those are solutions that we're going to see more come our way, which is fantastic, because that's going to help us continue to push the bar on what's possible. I do show nutrient demand here if you can't see it, but pulses, peas specifically, are a crop that needs a high concentration of nutrients for a long period of time, all the way out to basically reproduction. And one of the big ones, we go back to the total demand of, say, something like potassium. I think off memory, I think it's to grow 100 bushels, that crop needs to find 275 pounds of potassium on acre through the growing season. And so if you got, sandy soil, coarse textured soil where your potassium levels are lower, that definitely could be a limiting factor. And the plant is thinking every single minute of the day. It's sending roots out, and those roots are pulling back information to the DNA to make decisions to say, what should we be? And if it's not finding the nutrients, it essentially is then going to set itself up for essentially lower potential. And I learned this from Mike, so Mike's nodding his head, but plants.
I'm amazed. I love this.
I'm learning, Mike. I'm learning. Yeah.
This graph is what I use all the time, and everybody should recognize this, that the plant builds this vegetative, all plants grow like this. They have this vegetative growth, which is growing not just the leaves, but all the reproductive parts. And when it does, this flowering starts. And the trick with peas Of course, peas are somewhat indeterminate. They bloom over a long, long period of time. So if you're going to want to put in nutrient to load this plant, you're going to want to put it in before you get to this blooming period to load her into the plant so that it can use it to support the growth and the final production of the whole reproductive system, including the pollen and the anthers and all of that kind of stuff. So, you know, if you go in late, you're behind the ball. You got to, if you're going to grow this crop, you got to keep the pedal to the metal right from the get-go, as far as I'm concerned, on all aspects.
Yeah, we do have quite a few questions around the seed and germination. And one thing we will do is, you know, this very intensive study we have on the go right now that's basically cross-correlating nutrient content to vigor and germ. You know, we'll produce that report so everybody can have access to that. But, you know, I do think we are onto something that's, you know, if you grow a crop on a poor fertilized field, ultimately, to Mike's point, the seed's probably also going to have a lower level of nutrition. And when we talk about seed growers, you know, if they're the leaders in the industry, which we expect them to be, and they usually are, you know, we want to work with them to ensure that they're producing the highest nutrient dense seed, not just high germination, but to ensure the vigor is fully optimized with high levels of phosphorus, zinc, manganese, nutrients that are important to that. So it's all part of this process, everybody working together to figure out, you know, how to reduce the hurdles that take away from potential. We'll get into this key aspect of production when we talk pulses. That's obviously, you know, fixing nitrogen. In this case, you know, we're talking hundreds of pounds we need to find of nitrogen. And this is where Ryan and I have had this emotional connection over the last 17 or 18 years of our relationship, where you know, we've always connected on a deeper thinking of the soil health aspect and trying to learn more how does this relationship work with plants, soil, and biology. And I know Ryan's had a huge passion to understand soil at a greater depth. And so when we look at the importance of nodulation and the impact that this actually has on peas, and I know Meghan's done a fair bit of work and actually research for us because we're in this space as well. What are some of the key factors you're looking at here, like from a soil all the way through to biology in this relationship?
Well, when it comes to soil biology, I find it's one of those things where the more I learn, I don't know when it comes to soil biology. It is a huge space, and there are all sorts of different types of flavors of inoculant. We haven't done a lot on peas, but we've done more on soybeans. From that standpoint, we're looking to do more with peas. I think there's a lot to learn there, and with faba beans as well. You know, there's been a big debate about pH as well with that. Meghan, maybe you can talk about this. We've looked for white paper data on this, and you can maybe touch on that as well. Like there's a huge debate about this, and you go ahead, Meghan.
Well, we have a lot of higher pH soil in the valley, and it's kind of been told to us that high pH soils don't get good nodulation. And you know, we're pulling off some pretty decent pea crops and soybeans as well. And so Ryan had the question of, you know, well, let's compare this. How does nodulation look in different pH soils? And so we collected some soil in the fall. So we had down to a 5.8 pH up to an 8.5. So we did 3 kind of a low, medium, high pH soil, and we grew out soybeans, peas, and fabas in those soils in our grow room over the winter. It's just wrapping up now. And for the peas, I mean, we really didn't get good nodulation overall, like across all the soils, and I'm not exactly sure why. You know, it was our first time trying to get things to nodulate in the grow room, but the point was there was no difference in the really high pH. There was no significant difference in how those plants responded to the inoculant. So it's something that we'd like to continue in the field and see how that looks and maybe redo it again with on seed. We just used granular inoculant for this trial, but looking at some on seed inoculant as well. So
we've also looked at what. Go ahead.
I was just going to maybe to a point. I just wonder if there's some correlation there where the rhizobium bacteria for pulses is more native to Western Canada. Maybe they've actually adapted themselves over time to more accommodate a higher pH soil, where Brady rhizobium for soybeans is not native, so it maybe hasn't been able to adapt to our soil type as effectively. It's a thought.
Yeah.
I don't know if it's
could for sure. Yeah, it could be. Yeah. In the Fabas, also a native bacteria, but there was excellent nodulation across all soil types, like hundreds per plant. So that really the pH didn't seem to affect that at all. So
yeah. Now I do know there's visual nodules in what looks to be nodulation, and then the other side of that is actually nitrogen fixation. So,
yes,
to understand how effective those nodules are is really the other part of the conversation.
My staff's listening to this, so I should do a shout out to them. But yeah, of those 200 nodules on every faba bean plant, they're slicing them in half with scalpels to see if they're pink and active or if they're green and not doing anything. So I'm definitely considered that as well. Yeah.
That's perfect. Yeah.
So faba bean nodules are similar to soybean nodules, and of course, as you know, or maybe folks don't know, faba bean is about the best nitrogen fixing crop we can possibly grow. It fixes more nitrogen than any other legume that we grow. It is a great crop. I've spent 40 years looking at Lagus bugs in faba beans, but it is a great crop. For you guys that grow it for the first time, it's your easiest way to find out if you have pea leaf weevil around, because those weevils can sniff out faba beans from I don't know how miles away. And if you're going to find pea leaf weevil feeding on anything, it'll be on a faba bean. You can use that as a sentinel crop if you want.
You had something to mention, Ryan, and then I'll get moving along.
I was just going to make a comment on faba beans as well. They're the one crop, more so than peas or soybeans for that matter, how they can actually change the soil structure of your soil. Like, you can take some heavy Peguis blue clay and seed faba beans in there, and time after time, the customer has said to me the next year when they're cultivating or seeding, this is the most mellow I've seen this dirt in my life. Like, it's a common statement from every customer that's grown soybeans. It's actually quite remarkable what it can do to your soil. So,
yeah.
It's funny, Ryan, when I first started, I had asked some farmers, like, why are what's the benefit of soybeans or fabas? And they said, oh, it mellows out your soil. And I'm like, I don't know what that means, but okay. And then we had the research farm on some faba bean ground, and I was like, oh, this is nice. Like, I get what this, it was a huge difference. If I could follow fabas all the time, I would.
Yeah. So yeah, maybe a whole other benefit to a rotation. And I truly believe this is actually where this relationship between plant, soil, and biology come together. You got a high nitrogen fixing crop, piles of nodulation. It's allowing plants to exude a high level of acid. I think it actually is a real good soil builder, not just structurally, but also biologically speaking. And I think we're overlooking some of those things moving forward. Fabas in the rotation could be a real soil health piece to actually help with biological activity. And I will reference it here because there was a question from Darcy, and Darcy, you're asking some good questions today. But peas on canola ground, is that okay? And we would say absolutely. Now, you got to be cognizant of the nutrient demand of canola and then how much nutrients peas need. How do we fertilize that? The other side that we look at ourselves is actually the importance of mycorrhizae to pulses, especially peas, fabas, and all pulse crops. But when you grow canola, it's not a host plant for mycorrhizae, so your mycorrhizal populations actually deplete. They starve off, and canola actually even emits a toxin that kills off the spores. So including mycorrhizae onto your inoculant option for peas is important. And Ryan and Meghan's researched this, and Ryan has seen a lot of this used in the Swan River Valley on pulse crops, especially on canola ground. I call it an absolute. If you're growing peas on canola, you better bring mycorrhizae back into the equation.
Yeah, that and flax as well is a big one, I find too. So not that we grow lots of flax here, but yeah.
Right. Well, there's lots of interest in flax this year at $20 a bushel.
Yeah, there is.
Yeah. So yeah, our AgTiv line has mycorrhizal option in it, and I just say that's canola ground. It gives us an option to change the mindset on rotation. Peas on canola can be a great fit. Let's talk quickly about water. And I love the idea of understanding water in dryland farming. And, I think it's another phase of learning in agronomy that, we're going to dial in, and there's lots of companies that are focused on this. But, when we stack up peas to our core crops we grow here in Western Canada, it is only falling slightly short to canola as far as how much water it takes to grow a bushel. 3,770 gallons to grow a bushel. And I took this was from our last podcast, but I did a little more research to say, well, how many gallons of water is there in an inch of rain on an acre? And as smart as I am, and as smart as everybody is today, you can Google it and it'll tell you. And so I Googled it, and it actually told me there's 2,700 gallons of water in an inch of rain on an acre. And so you do the quick math, that's potentially 7 bushels of peas in an inch. And we'll, as we wrap up here today, we'll look at some of the research Meghan's done, where she actually, measured the rainfall every year and the research plots, and how that could actually correlate to overall production. So we talked dryland farming, a lot of peas, a lot of lentils are growing where it's drier. And yet, if we're talking 100, you do the quick math. If we get 7 bushels out of an inch, how many inches of rain do we need? And we need 4 inches to build the factory in the soil.
Yeah.
It's big. It's a lot of rain to grow 100. It doesn't happen easy. And yet, our greatest concern with rain and peas is what?
Wet feet.
Disease.
Wet feet and disease. So it's this conundrum that we got. We want to get there, but we gotta try to manage that water. So again, I'm going to come back to water infiltration, soil health. If you got ponding, chances are you're setting yourself up for some level of disease that's going to come into this crop. This is we talked about weeds early, but this is information I got from Meghan that she got from Saskatchewan Agriculture. I just threw this in here, but again, the importance of weed control early is something that we did talk to. It's a very poor competitor in its early days. The other challenge we have is, I say this on pulses, 30 days in or 25 days in, you're spraying herbicide to control weeds, which is, Ryan says, is critical. But you also have a plant, like Mike says, is taking a lot of its energy to try to build nodules. So you hit it with herbicide like a Viper, to control the volunteer canola. What does that mean for a plant? It takes a pile of energy for nodulation, takes a pile of energy to metabolize the chemistry in Viper, and yet we're still trying to grow a factory. And so I say plant health here is actually critical, and that's, some nutrition in the tank with herbicide. If we're talking 100 bushels, I just see it as an absolute must, where we got to keep the plants actively growing. Probably in all reality, we need phosphorus. We need phosphorus and potassium. We do have a product active VPR that, we're focused on plant health to ensure that, we don't stall out nodulation or stall out the metabolization of the chemistry in a Viper or an Odyssey or whatever we're actually spraying. So something to consider. Don't just think of killing weeds. I think let's focus on health to ensure nodulation happens early. We're not setting that plant back for 4 or 5 or 6 days. And lots of people have seen yellow peas after herbicide, and we don't like to see it. Especially if we're trying to grow 100 and that crop is done in 90 days, and you take 5 or 6 or 7 days where it's yellow. I say those are days you don't you don't get them back. Yeah.
That raises a point I want to make, just a reminder to everyone listening, is with these drier conditions we've had over the last couple of years, don't forget about flucarbazone rotation. Like if you're going after wheat and if you used Everest, it's really never been a big issue with our higher organic matter and heavier soils up here, but I've seen it. And it's something that we've kind of ignored or not paid a lot of attention to. But remember, if you have Everest, if you sprayed Everest or flucarbazone of any kind, and you're going into peas, it's something to be aware of, and remember that it'll curtail that 100 bushels pretty fast, for sure. The residue. So
herbicide rotation seems to be really important in growing peas and lentils.
Yep.
A lot of damage.
So we get to the reproductive stage here. Obviously, a critical phase again, flowering. Maybe talk a little bit about insects. I mean, obviously, Mike, this is your career.
Yeah.
Ryan, from a scouting perspective, how often are you finding pea aphids in peas at flowering?
You know what? Aphids here haven't been something that we've had to spray for in the valley in peas. It never seems to be too bad. I'm more worried about pea weevil. I've been finding lots of adults in the fall in the trash. You know, it's something to be cognizant of or be aware of, the fact that they're here and they're in the valley. So haven't seen any major damage, but why let it get to that point? Why not try and, ounce of prevention is always, better than a pound of cure, right? So if we can kind of get ahead of it, and there's seed treatments and things that can curtail that as well. So just be aware of it for sure. Yeah.
Yeah, and one thing that we do know about peas is that killing the adults or foliar, spraying out the adults doesn't seem to provide much protection. Each female lay 1,000 eggs. They just spray them on the ground. So basically all you're doing is revenge killing and making yourself feel good that you're killing a bunch of bugs. But if you're going to do anything, use a seed dressing, because what the weevil adult just chews on the leaves, it's the larvae that go down and live right in the nodule. They use the sugars and the carbon that the plant gives to the nodule and the nitrogens that's being fixed as their energy source. It's a great lifestyle. So it's protected in that nodule. So it eats the nodule and you lose nitrogen fixation and suppress the plant. So just don't waste your money spraying the adults, basically. If you're going to do anything, use a seed application, a seed treatment, and away you go.
And another thing too, there's you have a conversation with your farmer, your producer, and the conversation always kind of steers to, why don't I have that insect? Well, if my advice would be, if they're in the area, then it's probably a wise thing to try and prevent. It's harder to get rid of something that once you have it. I find just try and prevent it as much as you can. If they're not known to the general area, absolutely, you're probably fine. You wait and see. But if they're starting to show up, then it's probably good practice for not a lot of investment to prevent these insects. So
I'm going to throw down a thought, and it's probably controversial, but it's my 20 years of looking at soil. 20 years of looking at soil tests and tissue tests. Mike references the importance of tissue testing to ensure we have optimal nutrition at this critical phase of a crop's life. I would argue, there's going to be a correlation to concentrations of potassium in the pea plant and the infestation potential of something like aphids. And so we look at this over the years, aphids being a leaf sucking insect, it could be an indication that we are actually low in potassium, and that's why they're attracted. We've seen that for years on potatoes, where we do petioles every week, and they could always predict leaf hoppers before they actually came in, basis concentrations of potassium in the plant. So I'll throw that out if somebody wants to challenge me, by all means, but I always say there's a correlation between insects and plant health.
I don't think I can disagree with you, Craig, at all. I think there's more to learn about that as well, but I think there's definitely a lot of merit to what you just said. I believe that.
Yeah, so a tissue test could be part of an insect monitoring program as well, potentially. As crazy as that sounds.
It can be, and you can rectify some of that through foliar, but if you're if you're not tissue testing and putting on foliar nutrients, you're kind of maybe missing the target. How do you know what the heck you're really needing in the plant? So I'm a big believer that tissue testing is the blood test during season and the way you go with that.
Yeah.
Go back one, Craig. I got to make a comment about this for guys about aphids and everything else. Here's a pea. Inside that clam leaf, you have growth of other clam leaves, and there's a whole series, about 3 or 4 of them with every clam leaf. And then you get into, this is where the growing point is. So that's where we have to get nutrients, and all the new cells are produced there. But the next couple of slides are going to help you a bit. When the plant, a pea plant is an indeterminate kind of plant. So at every peduncle, you get 2 or 3 pods that form. Each one of those are in series. So, you know, and within each flower, they're in series as well in terms of the growth within that set of 3. So here you can see this one, the middle bottom one, where you can see the anthers are formed, and the next one is a close-up. Now you can move onward. But all of these here are just growing more leaves inside of that. So here we have a pea, and this is still inside the flower, and it's pollinated, and you can see all the pollen on the stigma, and it's going to grow down all of these pollen tubes down to pollinate every seed. And if it doesn't get pollinated, you don't get a seed there. Now, because there's a sequence in every little clump, and there's the first ones are on the bottom, and then they're higher and higher up. You know, peas can bloom for a prolonged period of time. So as the plant reaches that peak in its growth curve into flowering, the first flower is formed, get the majority of the nutrient from that plant, because the roots are still performing, they're still working. But as time goes on, the plant will start shutting down the supply of sugars to the roots, because now the sink is the seed. And that's what the plant is focusing on. So you got to get that plant built up to that particular time, and you have to have all that pollen ready to go.
Yeah.
So that's sequence. So you got to start early. You got to have, you know, if you're going to go on it with anything, the plant is sort of loaded at the start of bloom. If you want to support it for 2 or 3 weeks through that bloom period, you're going to have to add something in there if you don't have enough to carry it right through to fill those top pods. If you get what I mean? Because it's a sequential event.
Yeah, I would argue there isn't a plant that we grow in Western Canada, because we only got 100 days. If we can't build a factory early, and I'm talking the first 40, 50 days of its life, you know, how do we grow roots, get the factory built? Because when you get to reproduction, you're relying mostly on the factory. And sure enough, yeah, you can apply a foliar. I mean, in this case for us, active flower with nitrogen, phosphorus, potassium in it, and some boron. It's really just to try to help finish this reproductive phase of a plant's life. And, you know, it's been very effective on peas over the years. But you better
go ahead.
Yeah.
Sorry.
You better build the factory early, because Mike's talking having that nutrient loaded into the plant to support this critical phase.
Now think of it this way. If you've got the factory built, you now need the water to move everything into the seed. So if you get that dry condition at that period of time, the plant is going to really, really struggle. First of all, the plant has to build the seed in terms of cells. Then it's got to fill it with all the nutrients from the rest of the plant to complete it. That water at the end of the life cycle is pretty damn important. And last year, canola across the prairies got hit really hard because it turned hot and dry during that pod filling period. And guys were really disappointed in a lot of crops. A lot of people, you know, we want that water, but we need it throughout the whole life cycle of that plant, right through, except once it starts senescing. And I'm hoping your water stuff later on shows some of that in the year where you had poor yields.
Just so you know, we're at an hour. The conversation's been great. If you want to stick with us, you know, we still got most people on, and give us another 10 minutes, 15 minutes to wrap up. We got some great research material from Meghan here at the end that we want to get to as well. So we appreciate you staying on, you know, and conversation has been enjoyable here for me. So Just to mention, and this is probably on everybody's mind, but disease. When we talk about peas and even lentils, and then Meghan's going to show you some stuff even on Fabas, how impactful disease can be. But any recommendations, guidelines? You know, Barry's asked a good question here about any forecasting tools for disease that we could potentially leverage in peas that would help us. In this case, I think I'm showing Aphanomyces, but let's have a quick conversation and then we'll get into some of Meghan's stuff.
Well, my opinion on Aphanomyces is kind of scary. Something you definitely don't want. It's like the club root of peas, really. And the problem is, that's kind of, but worse because there's no genetics that we have available right now to prevent it. There's some seed treatments now coming out that might help a bit, but and it needs to be treated the same way as club root. Clean your equipment. It's soil borne. It's a scary disease for pulses. It worries me actually quite a bit, but the rotation and how the half-life of this is what I think Meghan is like 8 years or something.
They're saying 10. The spores, the resting spores, are 10 years viable in the soil. Recommending a 4 to 5 year rotation between pea crops if you don't have Aphanomyces, and if you do have a positive test in your field, then you should be more like 6 years between pea crops or pulse crops.
Yeah.
Because lentils are also affected. So yeah.
So there is an actual test. You can test your soil.
Mm-hmm.
Yeah. Okay, that's good for people to know.
Yeah, there the Manitoba, Alberta, and Sask Pulse Growers Associations put together a really kind of nice little handout on Aphanomyces. In 2017, they did a survey across the prairies. There was only 31 fields tested in Manitoba, but 45% of those fields had Aphanomyces present, which in Saskatchewan was 40%. So we're already pretty high.
And it's been dry. And I think I have my fears on a year with more moisture. This could show up. I don't want to be a doomsday guy, but I mean, it's something to be cognizant and aware of. Like, it's a serious thing that if there's any products, there's a product. Syngenta has a product too that, a preventative product. It's not perfect, but I mean, it's out there. These are things to keep in mind. Like I said, an ounce of prevention per pound of cure. So,
Another thing to mention is that they do suggest that if you want to keep a pulse in your rotation, that faba beans are a good option. They do have a partial resistance to Aphanomyces. So I'm just sharing what I've learned from the pulse growers, but that's at least a little bit of hope.
Yeah,
I think Meghan might want to talk about just on fungicides in general, some of the stuff we've done. It's been no less than positive, even on dry years, on some of the research we've done with fungicides, not just for diseases in general and peas. So.
Yeah, we've been working with the Manitoba Pulse and Soybean Growers Association through their on-farm network. So we have producers that are willing, and I'm very grateful to them that they'll do these field trials. It is a lot of work, and I do appreciate that they work with us. But over the last 3 years, we've done 9 different trials with peas. 6 of them were in fungicide, and even in those 3 dry years from 2018, 19, and 2020, 50% of the time there was a significant yield increase with a fungicide application. And this is many different products, different timings, 1 app versus 2 apps, 2 apps versus no apps. It all plays out in the year of looking at, okay, well, how much rainfall did we have? Do we want to go early? Do we want to go late? So it's not a really great comparison, but if you think you're going to need a fungicide at a certain point in your growing season, there's a 50% chance that it was the right decision, even on a dry year. So to look, if we had done these between 2014 to 2017, these results could be like, who knows what those significant levels would be. So Brian and I were talking, I was like, what do you need to do for 100 bushel peas? Well, you need a fungicide application. That's probably something you should bank on. But
a lot of guys are putting on 2 fungicide applications right now, even at whatever they're going at, simply to keep that crop from going down on them and keeping that flow of nutrients and water for transpiration to keep moving up the plant.
Mm-hmm.
So if you're shooting for a big yield, you're going to be watching that and monitoring that pretty closely and be ready to go for sure.
Yeah, and in this space too, in ag research, and if doing this, and we find that 50% is a really good number on a dry year. Like it's you feel confident, and there's lots of gambles you take every year, and this would be definitely one of them. Like for sure.
Yeah.
I should say that like 50% of the time it was a significant increase. 100% of the time the average yield was always increased, but just with the trial layouts and when you apply some statistics to it, you want to be sure that your differences are due to the treatment and not due to where you place the strips in the field. So you're not going to lose anything, I guess, by putting on
50% of the time it works every time.
Well, the good thing is there's a lot of great fungicidal options for pulses. I mean, a lot of the companies have brought forth some great products that give us that level of protection. And and then let's be honest, if anybody's tried to run through 80 or 90 bushel pea crop at flowering, good luck. You're going to get about 10 feet in. So you know the mass of material there, and you say, well, how is disease? It's a totally different environment in that canopy, and you say, oh, it's thick, it's dense, it's woven together. You say it definitely creates an opportunity for disease to actually take hold, right? So it makes nothing but sense. Yeah. Meghan, we're getting into some of your stuff here as we wrap up, obviously, and again, we appreciate everybody sticking with us as we've been over. But this is some of your variety trial work that you've done over the last 3 years, and I actually, when you sent this to me, I'm like, holy jack, I can't believe there's that many pea varieties out there, in the marketplace. We hear 2 or 3, but
there's, I think 20, I think I counted there was 19 or 20 that we've had all 3 years. And then on this graph here, there's a few stragglers that we've had either 1 or 2 years that we've looked at. But the point of this wasn't to show the actual yields of any of these varieties. I mean, they're all suitable for different areas, I'm sure. But just the trend of the 3 years that we did grow these varieties in that 2018 and 2020 were quite successful years for peas in our area. We're pushing, we had individual plots that were well over 100 bushels an acre when you factor that out. But on an average, we're pushing that 100 bushel mark here. But so when we were talking about this, Craig, and you said, well, what was the difference in 2019? So we went into the soil tests and there wasn't really anything that stood out to me that, if you're watching, there's the soil test of the top 6 inches of the profile there for the 3 years. So these are 3 different fields, different soils, textures. Nothing really stood out. We're like, oh, well, we were short in boron in 2019, and that's why nothing popped out at us. So then we looked at, the nutrients that were applied were roughly the same, and what it really came down to. So in 2018, our average bushels per acre is 84, 2019, 47, 2020, 87 bushels an acre. But in 2019, we had half the rainfall. And I think what's more important is that rainfall came too late. In 2018 and 2020, we had a really good soaker in June, and the crop needed that to build that factory. And in 2019, it just wasn't there. It came too late, and the crop suffered.
Yeah.
Is there anything in there, Mike, that you see that you'd pull out that was in the nutrient side of things?
No, but here's the problem. Because you had no water, what's in the soil is somewhat irrelevant. What you needed was the tissue test through those 3 years to tell you what the problem was.
Right.
Because, we know with potassium, we know with many of these that are immobile to a large degree, and potassium is really key. N and P build the cells. The potassium and water are what grow and expand the cells. We know that under dry conditions, things like boron, potassium, zinc, all those micros that are metals are going to be deficient. So it doesn't matter. But under water conditions, when you have water, the roots can grow and they can access that. They can't when it's dry. So that's the beauty. For me, as long as I'm with Taurus, whenever we do a trial anymore, I want a soil test, a tissue test, and a seed analysis done on that trial. Because you go from seeding to yield, you have no idea during that growing season what factors were affecting it. Just that simple.
Yeah, I would say the one that jumps out to me is obviously water. The 1 year that yields were down 50, 60 versus 80, 90, 100 is, you're missing essentially 4 inches of water, which if I do my quick math, 4 inches of water is 28 bushels a piece. You go back to your chart and say, well, that's not far off, right? When you look at the years. So as much as we are scared about water with disease, if we're talking high yield peas, we need water. There's no way around it.
It's almost worse than that, because 4 inches of water in one fall is not the same as an inch of water every week for 4 weeks.
Right.
Sure. Yeah.
So even there, you got to be cognizant of when it comes and how much.
Yeah. In 2020, that 295 millimeters of rain there, we had 4 inches of that in a couple of hours. So it probably is more like 200 for the season, right? So.
Yeah. Just a quick, we're going to wrap up here pretty quick, but there's a question back to the fungicide. Obviously, is it something that it adds yield? And I'll say straight up, no. All we're trying to do with fungicides is keep the yield that we started with. We're trying to salvage and protect. Does everybody agree with that?
Yeah, for sure. Yeah.
I would think so.
100%. Okay. Fungicides do not add yield. They keep the yield we had. But the concern here in this question is, does it actually prolong maturity?
Yeah, it can too as well. I believe it does. It's just that it's a healthier plant and it'll live longer for sure. But with hand in hand, if your plant's going to live longer, you're going to have seeds filling, pods filling longer. It's going to equate to yield. So
your nutrient supply is going to affect your senescence period, and then mostly nitrogen is responsible for that prolongation, if you like, of the green period in plants.
I wanted to. I don't know, I'm running out of time, but I wanted to do one thing. I just wanted to ask Mike too, we want to do some work with protein in pulses, because it's a big thing right now. And I just wanted your quick snapshot on what are some, and maybe even to the audience listen to this, what are some things that you think you might want to try or do to try and manipulate protein in peas? Meghan and I are coming up with a few ideas on low-hanging fruit is nitrogen and sulfur, and we're thinking about maybe doing some foliar urea trials at certain times of the plant life, just to see if we can actually try and increase protein or manipulate it to some degree. But what would you think, Mike?
This is a question that's been coming up for us in the last while, and I've been digging through the literature over the last couple of weeks, and I can't actually find a whole lot of this, to tell you the honest truth, out there. Definitely, N-to-S ratio has got to be a major factor. But the plant, the plant can access things quite well, and I think it's water. I mean, we know more about wheat. When we get high protein, when we get poor growth, and we get a lot of nitrogen. Well, with peas, it's already a high protein crop.
Right.
30+% protein. So it's tough to move that up big time. And the genetics, there's got to be a genetic factor for sure between varieties. It would be the first place I would look for that. Then I would look at the balance of N to S and maybe move that up I would also then drive my phosphorus and my potassium as high as I possibly could in there. And that's about all I could do. My micros I wouldn't be worried about. If I didn't have that phosphorus and potassium first, along with the sulfur and nitrogen, you know what? I'm a believer if you don't have those in place that make the larger plant, the micros aren't going to save you.
Yep.
You know, and the timing of that is critical too. Like if you're going to put a dribble on with the nitrogen, you want to blast mid-bloom or somewhere, and I hate doing much during the bloom period, to tell you the honest truth, because that in fact stresses itself. But if you can dribble it, or you can put a delayed nitrogen application on in some slow release mechanism going into the end of the crop, I think that's where you're going to drive her up. But it's not clear to me, Ryan, not clear to me how we drive that protein higher.
Yeah, if you want my thoughts, I would say let's leverage the tools we currently have. I mean, I don't really know if nodulation or using biology can get us 100% all the way there. You know, that's why we're talking about maybe top dressing nitrogen, which I think there's some merit to. But why not even on peas? Or let's talk about double inoculating. We're talking 100 bushels. If double inoculating actually helps the biology do more of a job or find, you know, extra nitrogen for that plant, more nodules means then we better have more other fertility to support those nodules too. But, you know, that's one thing that, you know, Ryan's looking at this year in a lot of their pulses is double inoculating, putting a, you know, inoculant on the seed plus an in furrow. And I think that's going to be another tool to help us not just pursue yield, but make sure we got enough N. I mean, and I say we can never have enough sulfur on our soil. So, you know, back to my days at Tagger where we were broadcasting 100 pounds. I mean, we see a lot of that going on today still, and even more so today with biosol and things like that, you know, let's tighten that N-S ratio as tight as we can possibly get it, because that's only going to help us when it comes to trying to derive protein at the end of the season on a big crop, right? So
these are nutrients that are easier to remedy in the soil too. They're easier to deal with year to year, right? So it'd be a quicker fix if you could identify if there was an issue, right? Like I find or think so.
Yeah.
I think that would be a great research project for you guys to take on, is to look at that, because it's a significant question coming up all over the place, and it's going to stay here.
Yeah, we want to. We would be open to ideas too. If anyone can think of something that they'd like to try, we have the resources to do that. So
we should talk.
Yeah, we have a survey at the end. For those that have stayed on, we'll send out a survey if you got some ideas on what you'd like to see, specifically for Meghan and her research initiatives. You know, I like this conversation because we're talking about a human protein crop here now, both for Fabas and peas. Protein, I got to think for the processors is going to be important. You know, they're going to tell us what varieties to grow, but let's figure out how to optimize protein. One last thing I'll mention too is Meghan has done some research for us on soybeans, looking at the impact of protein when using mycorrhizae. And we have seen, you know, some increase in protein on soybeans with the inclusion of mycorrhizae into the biological application up front as well, which is to me, that's phosphorus, finding more phosphorus, which builds more energy, which gives us a chance to have a stronger plant.
Sure. It's going to be a question. I think we know already that genetics have a big effect. We're seeing some of the protein plants having some favored varieties that they preferred varieties. So I think that really alludes into the fact that they're seeing a genetic difference with protein. But yeah, like Craig you just said, there's a lot of things that can come into play. But I believe that there's a reason for everything, and we can figure it out. You know, we can. You know, it might take some time, but yeah.
I think that's your best chance on a short run is the genetics, to tell you the honest truth.
Yeah.
Until we figure this out.
Yeah. So what it takes is it takes people that are on this webcast that have passion to find ways to learn and take the blinders off and say what is possible. And you know, I think the conversation today has been fantastic. There was no way we were going to fit it into an hour, and so we've gone over time. And I appreciate when everybody contributes. I mean, this has been great. I'm learning myself. I always do. That's why I enjoy doing these podcasts. And, so to wrap, we've covered everything from, having good balanced soil, obviously drainage is critical, long rotations. We got to think fertility versus fertilizer. Leveraging this biological activity that we have for pulses is key. It's a huge asset that we have in these crops. Water, as Meghan has identified in her 3 years of research on peas, is critical. And in plant health throughout, weeds, as Ryan says, if you don't get them, you're in trouble. They take nutrients and water too, right? So, and pick a variety that's best suited for your area. Megan's identified that some varieties have performed better in the valley. There's no question, right? So, so with that, any closing comments that you want to cover off as we wrap?
I would just say this has been good. I was a little nervous my first podcast, but we settled in pretty good, didn't we? And we kind of almost the redheaded stepchild of the pulses, faba beans. We never got a chance to talk. Never got a chance to talk about faba beans much. We could probably talk for 2 hours about that alone, but that's a passion of mine too. Faba beans. There's so much to learn about them as well, and they're pretty cool plants. So yeah.
One thing we didn't mention, but Ryan and I had talked about it. You see it a lot because your peas are coming off in August, and you see those beautiful pea crops in September. And I think some low-hanging fruit is set your combine yourself the 4 or 5 bushels that you're throwing out the back. That's just an easy thing. If we're trying to push it and we want a product, and those products are going to give us 3 or 4 bushels, well, take the time and set the combine, and there's 5 bushels for you. Pretty easy. I say that easy, not ever having set a commercial combine, so I should maybe check what I'm telling you guys to do, but seems like a good place to start.
Yeah, there is I think a good tool out there, Bushel Plus. It's got a pretty good tool to actually help us manage combine settings. And I agree with you, Megan. We do all this stuff, and then we throw 5 bushels at the back. You say, well, who's fooling who here, right? I mean, that's $45-50 worth of crop that we give away, right? So
those guys don't like using that tool. They don't like to know what they're throwing over.
Exactly. Right. You don't want to know.
Yeah.
Well, great. You know what? There's a couple more questions. We'll try to get some here quick. Mike, tissue testing. What's the proper timing on pulses? This is from Barry. Barry's asked some great questions today. Thanks, Barry.
He is. You know what? If I was going big time, I'd be tissue testing about 6-7 node, or maybe even before. If I'm going with a foliar spray, I would be going before that spray so I could add any nutrients in a herbicide application. I would be going before 2 weeks before I'm expecting my first blossom to appear. Would be where I would go into that. I would be doing it on the upfront part, because the later you go, the less you can do in terms of really having an impact on the progression of this. And the one point I did want to make for many farmers, we talk about nodes on peas, and a lot of guys miss the timing because they don't count the node. The plant can be really, really small and have 5 and 6 nodes. And you think, well, it's just a small little plant, but it's already in the node stages. So you got to count the nodes. You got, I'm starting to say this, the harder you look, the more you find. But if you don't look, you're missing the boat.
Yeah. Okay. One last question. Lagus, concern on Fabas from John.
It's a big deal. It's important to monitor and watch, especially just after flowering, early pod. They can do a lot of damage, and it could cost you a lot of money because it's a big grading factor. Monitor closely. I did a whole webinar
on Lagus and Faba beans. I worked on 30 years ago before anybody even knew what was going on. They're very difficult to control in Faba beans, but I know exactly what happens to them, and is something we got to work on if we're going to have commercial Faba beans. Now, Saskatchewan, Alberta has real bad lagus, and I think you guys in northern Manitoba. Most of Saskatchewan doesn't have a big lagus problem, and that's a great place to grow Fabas. I
think it's just a matter of fact. We just saw all the canola we grow here. There's a lot of lagus around, so.
They seem to be more related to the Aspen Parkland kind of area, as far as I'm concerned. I mean, they outbreak from time to time, but they're more residential there. It's another topic.
Yep.
Okay.
We will have this podcast recorded, and I skipped over some of Meghan's slides on Fabas, which Ryan says the redheaded stepchild, but there's some great research on fungicide with Fabas. Gosling's asking about inches of water to grow Fabas in season. I think Ryan could speak to that. In a year with good moisture, the potential is significantly higher. They need water, probably like peas. I don't have that answer. Mike might be able to answer that after in the response section.
August rains, a lot like soybeans. It's a huge deal.
Okay. Yeah. So August rain for Fabas, like soys, if we get it, yields can go up 10, 15, 20 bushel an acre.
Great. Yeah. 100%.
Yeah. Okay. Well, with that, again, thanks everybody for coming on. I enjoyed the conversation, and lots of questions. We will any question we missed, we'll try to answer it after, and we'll send that out to the attendees. The podcast will be posted so you can watch it in its entirety if you're just listening today. So maybe look at some of the reference to the webinar slides that we showcased. So again, Ryan and Meghan, thank you very much. Your input's been tremendous, and we look forward to working with you moving forward. Hopefully there's some thoughts on protein that will come in the survey that you can leverage too in some of your research moving forward. So with that, thanks for coming on, and thanks everybody for joining us today and sticking with us for a little bit longer. So again, weather's improving, and get outside and enjoy it while you can before spring comes. So
great.
Take care. Talk to you soon.
Thanks, Craig.
Thanks, Craig.
You bet.